Can scientists really edit human DNA? Imagine this. What if doctors could remove a disease from your DNA before it even develops? What if inherited disorders could simply be erased? This is not science fiction anymore. It is called CRISPR, one of the most revolutionary discoveries in human biology. Every cell in your body contains DNA. DNA is the instruction manual that tells your body how to grow, how to function, what color your eyes are, your blood group, even your risk of certain disease. Humans have about 3 billion DNA letters packed into nearly every cell. Sometimes just one tiny mistake in this code can cause serious disease. CRISPR stands for clustered regularly interspaced short palindromic repeats. Fortunately, you do not need to remember the full name. Think of CRISPR as a molecular GPS and a pair of genetic scissors. It helps scientists find a specific DNA sequence and cut it remarkable precision. Surprisingly, CRISPR was not invented by humans. It was discovered in bacteria. Bacteria use CRISPR as an immune system to defend themselves against viruses. When a virus attacks, the bacteria recognize the viral DNA. They use protein called Cas9 to cut and destroy it. Scientists learned how to adapt this natural defense system to edit DNA in plants, animals, and humans. Think of editing DNA like editing a sentence. The cat sat on the mat. Suppose the word cat is wrong. CRISPR works like this. A guide RNA gRNA finds the exact DNA sequence. The Cas9 enzyme cuts the DNA at that location. The cell repairs the cut. Scientists can remove, replace, or insert DNA during the repair processes. It is similar to using find and replace in a word cursor but on DNA. Researchers are studying or using CRISPR for conditions such as sickle cell disease, beta thalassemia, certain inherited forms of blindness, some cancers, muscular dystrophy, cystic fibrosis. Several CRISPR based therapies have already reached clinical use for specific blood disorders in some countries, making a major milestone in gene editing medicine. CRISPR is not only changing healthcare, it is also used to develop disease resistant crops, improve food production, study genes in laboratories, helps conserve endangered species, explore new approaches to fighting infectious diseases. CRISPR raises important ethical concerns. Should scientists edit embryos? Could gene editing be used to create designer babies? Who decides which genetic changes are acceptable? As the technology advances, society must balance its enormous benefits with responsible uses. Scientists are now developing newer tools such as base editing, which changes a single DNA letter without cutting both strands. Prime editing, sometimes described as a more precise form of gene editing for certain mutations. These advances aim to make gene editing safer and more accurate. CRISPR have transformed biology by giving scientists an unprecedented ability to edit genes with precision. It offers hope for treating inherited disease, improving agriculture, and advancing medical research. Although many changes remain, CRISPR is already reshaping the future of medicine. Today's science fictions could become tomorrow's medical treatment. If you enjoyed this video, do not forget to like, share, and subscribe to Science Catalyzer. Thank you.